Okay, good morning. Let's have a class. We will be talking about the human disease caused by bacteria, the left section today. Now, before we go through some of the new stuff, let's review Clostridium. Because it's been a while, couple of days, and the Clostridium is a hot topic, so we want to review it. So Clostridium, it is gram-positive, spore-forming, large-shaped bacteria, and it's anaerobic bacteria. Okay, we talk about Clostridium difficile. Clostridium difficile, we also call it a CDIF. This is related to is antibiotic caused water diarrhea. And it is, you have a pseudo membrane colitis. And this is number one. The number two, what we talk about is Clostridium botulinum. And we talk about is infant botulism, wounds botulism, and the last one what we talk about is foods botulism. And this is typically for canned food, and we also call it a low acid food. Okay, that means pH 4.6. We will say more than 4.6, and water activity around 0.85. And the third one, what we talk about is Clostridium tetani. When we talk about the Clostridium tetani, it will cause lockjaw. So LOCK, lockjaw. And we also want to mention this, you have a DTaP vaccine to do the prevention. This one is also called a cause neonatal tetany. This has happened when the woman do the delivery. In the old time, they do not have a good hygiene system, operational surgery hygiene system. Then the neonatal tetany will be transferred to the newborn babies. So this one, I forgot to mention. The last one, Clostridium perfringens. Perfringens, this one will cause gas gangrene. This is a typical symptoms, and there is a way to do a little bit of rapid testing from the clinical standpoint. This is on a blood agar. Okay, let's say on a blood agar. You will see there is a two colony stacked together, and then they have a beta hemolytic zone, so we call it a double beta hemolytic zone. That's a quick test in the clinical area. Okay, so this is a Clostridium. We talk about that. Today we're going to move on to our last section. We will talk first of all is sexual transmitted disease. That is called STD. Sexual transmitted disease. We want to mention something to talk about. Sexual transmitted disease is a severe global health problem. You can see this is around 300 million cases globally. Globally, okay? And this is a disease is 95% is from male to female, Said very simple is infected or contaminated peanuts go to vaginal tract, cause a problem. It's all from there. And lots of them will be transmitted through a very soft mucous membrane system. So 95% from male to female. There is no vaccine, basically no vaccine to prevent sexual transmitted disease. Although you heard about there is something happens for, we can say HIV virus or something, there's some kind of vaccine comes out, but still we think there's no vaccine. And there is no immune memory capability. So what that means, which means if you get a syphilis, it does not mean your immune system will remember it and next time you will not get a syphilis. You get a gonorrhea, got a chlamydia, does not mean you get it once in your lifetime and the rest of your life you will not get it. You don't have an immune system. You could get it repeatedly. Last one, number 5 is about 80% of the time it is asymptomatic. So that gives you problem. People go ahead to have a sexual activity, continue after that, but it is asymptomatic for themselves. The last one, people always have a debate, is this a social problem versus scientific issue? This is always a big debate. We should be doing more education to solve them from the social education activity, or the medical science area should develop more progressive, more developing, more medicine tools, anything else to prevent or cure the sexual transmitted disease. So that is always a debate. Okay? Now the last one, condom use is always the most protective methods. Okay? So this is some of the outline, which means the opening remarks when we talk about all these. We will talk about 2 major sexual transmitted disease here. Number 1 is gonorrhea. Look at the picture, you will know. Neisseria gonorrhea. Okay, Neisseria gonorrhea. Since we talk about Neisseria, we mentioned about that before. Neisseria meningitis. So it's the same. Gram-negative. It is Coccobacillus. What the morphology? What's arrangement? Coffee bean shape. This is very typical. Looks like a coffee bean, like this. So we also call gram-negative diprococcal. Gram-positive diprococcal, by the way, is Streptococcus pneumoniae. So gram-negative diprococcal. Gram-negative Coccobacillus. What is Coccobacillus? Looks like a coccal, but it is a large shape, tiny large shape, and a coffee bean shape. That's a typical morphology and arrangement. This is a fastidious bacteria. What means it is a fastidious bacteria? We mentioned several times, if we have a Macanthi agar, and if we have a blood agar, and we have a chocolate agar, let's say, and this bacteria is very picky. They will not grow on Macanthi agar. So it will not grow on the blood agar, it's only grow on the chocolate agar. The chocolate agar is called modified Sayre-Martin medium. And it is also a transport medium. A transport medium. It's also need to go to the reference lab. Number 3, this bacteria we are talking about is the cell morphology. The colony morphology is very typical. What it is looks like. It's a pushable colony. And the last thing we want to mention, this is oxidase test positive. We will do this in the lab. Oxidase, during the electron transport chain, some of the bacteria using electron from cytochrome C go to oxygen. They need oxidase. In the test strip, we are showing you purple color, and it is oxidase test positive. Not many bacteria. Purple on test strip. Not many bacteria is oxidase test positive. So you can see it here. It is very typical colony. Okay, what are going to be the symptoms? The symptoms, you look at that, is urethral pus discharge. The pus come out from the penis. Also accompanied with dysuria, which means difficult to pee. This happens usually for male. Okay, for female, what's going to happen? Same thing. You have a urethral pus discharge from the vaginal tract, but you have a abdominal pain with unusual, which means on period time bleeding. This has happened in the female. Okay. Well, the gonorrhea at the beginning, there is some of the mild symptoms could be. You have a fever, you have a flu-like symptoms, but later on, it would be very invasive, will cause a cardiovascular disease, or maybe immune system will be damaged, or neurological symptoms, or could be developed. And this is a transfer through mucous membrane system. Okay. Now we want to talk a little bit more is about how we do the identification between male and female. There's a little bit difference between that. Little bit of difference is if a male will get a pus, then we're going to do the gram stain. Okay? If we find it's gram-negative, Coccobacillus, and it is a coffee bean shape, and plus we have an oxidase test positive, a grow on the chocolate agar, we will say this is presumptively gonorrhea. For the female, there's a little bit difference. Female, we do not do gram stain. We get the discharge from the vaginal tract. We don't do the gram stain. We skip this. Why? Because in the vaginal tract, there is a lot of the bacteria, lactic acid bacteria is dominant there, and some other normal bacteria will be bothering the gram results. So we don't do that. How we do it? We directly jump to strict plating onto modified chocolate nut. So we call it a modified cell multimedia. And we will directly transfer them, go to CDC reference lab. To do the identification. So a little bit difference between male and female. You can see Neisseria gonorrhea, what it looks like. Coffee bean shape coupled with white blood cell. It's a macrophage, these are white blood cell. It's a mucous membrane transferring. And that also could be because newborn baby have gonorrhea. What happened? Conjunctiveness of the newborn baby. So when you see the conjunctiveness of the newborn baby, it's either chlamydia or gonorrhea, which is coming from the mother. So just want to let you know, newborn baby could be one of the victims for sexually transmitted disease. And here on the top, you can see all of these things: gram-negative, oxidase positive diprococc cell, and attached to the microvilli, and photocytosis by the mucous cells. Okay, that is gonorrhea. Gonorrhea usually has a vaginal tract discharge, urethral tract discharge, have a pus, pain, burning, urination. We also call it a dysuria. And it's a pelvic inflammatory disease, results from the fallopian tubes and the surrounding tissue. And they could be very invasive, could be go to joints, heart, and the throat. Okay, just want to let you know. And how we do the diagnose, DNA probe test and a commercial test kit, oxidase test. What's the treatment? Antibiotics. But more and more we have the penicillin resistant comes out. And for the sexually transmitted disease, like we just mentioned, it could be asymptomatic. Public education condom use is more beneficial to be the prevention compared to the antibiotic therapy. Okay, because lots of the times when you find it, it is almost already being invasive. Next one, syphilis. Before we talk about this, we want to review some of the basic knowledge. You may still remember, you may forgot about it. We want to talk about, then we talk about the little bit detail of the syphilis. Okay. Move these out. So first of all, when we talk about syphilis, we are talking about is a group of bacteria, spiral sheets. We mentioned about that a couple of times. Spiral sheets is a special bacteria. This is a bacteria typically is very soft. And they are going to the body is three possible way is cuts, bites, and abrasions. And these bacteria, they do not have a very typical cell wall. There is no typical cell membrane system, cell wall, cell membrane system. So for example, this special bacteria, they do not have a lipopolysaccharides. There is no lipopolysaccharides. So there is no endotoxin. However, they have a very special lipoprotein will cause the toxin is going there. Okay? This is the bacteria's arrangements and the morphology. It looks like this. So we call it a spiral. We also call it asexual filaments. And we do not have a typical structure, like say flagella to do the movements. So we call it is a very similar name. We call it endoflagella for the movements. And we using a very special microscope to do the observation of spiral sheets. If you remember, is dark field microscope. It is different from the light field microscope we use in the lab. Okay. For spiral sheets, we are basically talking about the 3 examples. Treponema pallidum causes syphilis. Borrelia burgdorferi causes Lyme disease, and leptospiral interrogans, that's called jaundice. We'll mention these one by one. Okay. So first of all is syphilis, Treponema pallidum. Okay, so syphilis. Syphilis, pathogen spiral sheets, Treponema pallidum. Pallidum, sorry. Okay, spiral sheets we mentioned, use dark field microscope. Syphilis has a different stage. This is our first sexual transmitted disease recognized in the world. A long time ago, back in the Eastern Asian or the Southwest Asian, nobody can cure it. Okay, it's a big problem that killed lots of people. Obviously, the victims will be sexual activity person. There is a certain reason behind that. We'll talk about that. There is a 3 stage. First of all, called the primary stage. Primary stage, it is usually about 2 weeks to 7 weeks. The primary stage, we call it shanky or shankery. It is a French word. What is the primary stage looks like? The male penis will have red ulcer. That's for 2 to 7 weeks. You have a mild symptoms of fever and flavor. People doesn't recognize that too much. So moving on to secondary stage. Secondary stage for about 10 to 30 days called guma. It's also a French word. What is the guma looks like is a whole body rash. But the tricky thing here is, once you have a guma, it took about 10 to 30 days. This guy is gone. This is disappeared. So the people will say, okay, it's recovered. Let's go have fun. Then you showed up the whole body rash. People doesn't recognize things. It's maybe a skin disease. It's a mild skin disease. Is that right? It's possible. You use some of the ornament, maybe can recover. They think about. Because sexual transmitted disease is always from the mental area. It's not really the other things happen. So they go ahead to continue to have a sexually transmitted activity. What will happen? It will be transferred to the other person. Then we go to the last stage. Tertiary stage. This will be very tricky. This could be 30 days, 1 year, 5 years, and up to 30 years. People could carry on these spiral sheets the rest of their life, and later on will become neurological symptoms. That is a major thing for the problem. Okay? And the same thing, when they transfer, go to the urethral, and from the pregnant woman go to the newborn babies, they also could have a congenital syphilis. This is a picture of what it looks like. This first one is primary, small painless red ulcer. Look at the picture, that's called a chancre. The second stage we usually call it a guma. It looks like the back. This is the more severe one. Lots of the people who have the mild symptoms, you're not going to see anything. And the tertiary stage, guma usually in the second one. The tertiary stage is the skin, bone, nervous system. The lesions happens, and it could be have neurological symptoms. So this is the three major symptoms. The tricky thing is that we mentioned that the chancre will be disappearing. So people doesn't recognize that. And the last stage is that you have mild symptoms all the time. When you have 5 years, 10 years later, then if you continue to have a sexual activity, then will be transferred to the more people. Because it's asymptomatic, could be for 30 years. So that's a challenge for that. Diagnose microscopy exam, serological exam, the prevention, you can use antibiotic therapy, but not going to be very effective. So that's why we mentioned here is a public education, the new cases, sexual hygiene, use of the condom will be the more effective way to do the prevention. Okay? So that is for the syphilis. Remember these three stages. So that's to finish the first one, talk about syphilis. Treponema pallidum. Then we want to talk about is Borrelia burgdorferi, is Lyme disease. Lyme disease, the spiral sheets is Borrelia burgdorferi. Okay, Borrelia is a genus name. Burgdorferi is Burgdorferi is a species name. This is a scientist name. Dr. Bacterial is a medical microbiologist in Montana State University. In 1970s, he had a lot of the research and recognized this pathogen, this spiral sheets. This is coming from in 1970s in Lyon County, Connecticut. The first time comes out. That's why this is called Lyme disease. This is usually in the United States. If you look at the map, where it is, northeast, south, Midwest, and west. So in the United States, usually it's northeast, south, Florida area, east coast, and the west. Those are the risk area. If people go in there to do a deep diving, or like a fishing, deep ocean or deep marine fishing, they have to do an annual testing for the Lyme disease. What are the symptoms? The symptoms we mentioned about before is called annual rash, which is on the arm. There has happened is a target rash, like this. So we call it a annual rash, or we call it target rash. These are various typical symptoms. Now, the same thing like syphilis, all these stuff happened is mild symptoms at the beginning, but later on, they were developing fever, flu-like symptoms, cardiovascular disease and neurological symptoms. It is invasive. Now, where is this guy comes from? This is not really coming from the human to human. This is coming from a ticks. The ticks is special for the isodex ticks. But I tell you one thing, this is a long way to do. We talk about that. I want to draw the picture for you what happened. Okay, I usually don't look at the notes, but this one I have to draw the picture. So, what happened is in the environmental area, let's say there are ticks. Okay, the ticks right there. Isodex ticks. The ticks, you have an egg comes out. So a couple of eggs comes out. And this is in the spring season. A little bit warm, a little bit cold. Okay, when you have these eggs come out, what's going to happen? They're going to move on to, in the summer, they will have larvae comes out. Okay, larvae is like this. The larvae is actually infectious, which means they don't have really infected function. So we have the uninfected. You have those larvae. When you go to larvae, what's going to happen? Then go to the fall season. The larvae are going to be transferred everywhere. Then in the forestry, or some of the environments, you're going to hide this guy right there. You're going to find you're going to find the mice. So the mice will be there to eat these larvae. Then, going to the winter, what's going to happen? These fecal sample, the poop sample from the mice, will contaminate in those leaves. So you have a leafy there. These leaves will be contaminated with those larvae. Okay? And at this moment, the guys survive there is no longer a larvae, they will become a nymph. These nymphs will be highly transmitted. Then what happens in the spring? A human gonna come out, okay? Somebody gonna come out. Somebody goes there, okay? Do the swimming, do the diving, fishing. In those environmental areas, when they touch it, it's get contaminated. They will have these annual rash comes out. So typical target rash. Okay, then what's going to happen? This guy's fecal sample sometimes also go to the environment. No matter what you do, go to the hospital, do the treatment, go to the summer. These fecal samples usually go environment, which guy will be there. And I tell you one thing, when you have a fecal sample, you could have a male, and you have a female, and the male ticks comes out. These ticks comes out, okay? Then you're going to find what? Find this guy. Okay, I'm not sure draw very well. Try. Then we have this. You're going to find a deer. So, at the end of the day, what happened is the final dominant, the final host is a deer. Now, you cannot kill all the deer in the forestry, is that right? You cannot kill all the deer. You can do a deer hunting sometimes. So what do we have to do? You have to control that guy. They have to control the ticks. Then you go to next year fall. The winter, spring goes like this. So this is, you go from beginning to the end. This is a 2-year program, like a master's thesis program. At the end of, these spiral sheets will become mature and go to transfer for everywhere. So you can see that what the key here. There, the mice control will certainly helps, but in the environment it's difficult to do. The ticks control is key. And finally, actually, the dominant, the final host is the deer. So that's very interesting. You can look at here. That's a program. The same thing right here. The year 1, the year 2. Okay, so this is, we also call it isodex ticks cause the disease. It's a Lyme disease. It's very interesting. People think about it, it's just like human to human transfer. It's not. It's really from the environmental then go to the human being. Okay, so that's a Lyme disease. How to diagnose? Lyme disease, we have a ELISA testing kit and the polymerase chain reaction, the DNA testing methods. If we really have it, antibiotic therapy test at the early stage. However, more important is tick control and avoid ticks. Okay, so this is the key for what we talk about. Okay, so we are finished talk about here is this section we finished is about the disease of the bacterial caused disease. So I stop here, but